I don't have time to really tell you much about the biology of mitochondria themselves, but there's just a few little facts I want you to bear in mind because they'll help put the rest of the talk in context. So, I'm sure you're aware mitochondria are these tiny organelles. They're present in virtually all mammalian cells. They actually have quite a number of important functions, but the two that we're really going to focus on in the context of aging at least are ATP, or energy production, and then also ROS, homeostasis. I'm going to come back to that later. It's also important to remember that mitochondria have their own DNA. It's this compact circular molecule that you can see here. It's full of genes that encode for proteins of the mitochondrial respiratory chain, which is, of course, what produces energy within the cell. Just as a caveat, though, studies that have characterized the mitochondrial proteome have shown that actually there's probably at least 1,000 mitochondrial proteins. So clearly, those that are encoded by the mitochondrion itself are quite a small proportion of that total. And then the final thing to bear in mind is that cells contain multiple copies of mitochondria, multiple copies of this mitochondrial DNA, and that can range from a few hundred to many tens of thousands of copies per cell. And the implication of that is that if you have a mutation in your mitochondrial DNA, for example shown here in red, that's going to exist in a mixed state with the wild type, and we call this mixture of mutant and wild type mitochondrial DNA heteroplasmy. And then over time, this mutant mitochondrial DNA can come to predominate in a cell as shown down the bottom here, and then this is when the cell becomes sick. And we call this process of going from a low level of mutation to a high level of mutation within a cell clonal expansion. And that's something, again, I'm going to come back to later on. So mitochondrial aging is kind of a broad topic which we could have a whole 2-day workshop on if anyone really wanted. But, and this is, Julian's nodding his head, okay, so that's next year, guys. So look, this is a diagram I just found on the internet of the systems biology of aging, and as you can see, mitochondria up the top here in pale blue, everything's interconnected. So clearly, I'm not going to focus on all of that. What I'm going to do in the rest of this talk is just pick 3 or 4 questions which I think are pertinent to this meeting, which I think are also topical in the field at the minute, where our view of things has changed in recent years. Inevitably, this is going to be biased by kind of my own interests, which particularly around muscle. And I think it's also useful when you're reading any data on mitochondria in the context of aging to think, well, how kind of proximal to the mitochondrion itself is what I'm reading about. So I thought I'd start with aging phenotypes. We've heard a little bit about this already. I'm sure we're going to hear more about phenotypes such as frailty in the rest of this meeting. So how can you attempt to measure mitochondrial function in vivo in a human? Well, the best tool we have to do that is probably phosphorus MRS, magnetic resonance spectroscopy. So what you do, you have your patient lying in the MR scanner, you get them to do some exercise, usually exercising their calf muscle. There's a coil sitting on the leg which requires a spectrum which contains the various metabolites of phosphorus. So that's what's shown on the left here. And going kind of into the page in the z-axis, I guess, these are acquisitions over time at intervals of a few seconds. And I want you to focus on the big peak here labeled PCR. So this is phosphocreatine. And if you look at this graph in the top left here, the solid curve is what happens to phosphocreatine. So it depletes as you exercise, and then as you recover in the scanner, it's resynthesized. And how quickly you can resynthesize that is dependent on the quality of your mitochondria. So if that curve is slow in recovering, then that's poor mitochondrial function. So this was a study from a European group published just a couple of months ago, and they've done exactly that in the context of elderly individuals classified as either pre-frail or active robust. Now, I think the reason that they went for pre-frail rather than frail individuals is to try and see if these mitochondrial changes are already there in pre-frail, then it may be that the mitochondrial defect is proximal and is driving the phenotype. So if you look at the top left, first of all, panel A, this is exactly what I've been talking about. So this is the MRS, and you can see that it is slow to recover in the pre-frail group. The next several panels are all different mitochondrial proteins and enzyme activities measured in muscle biopsy, and essentially they all show the same thing, that it's also impaired. And then finally, mitochondrial DNA, so they've measured levels of mitochondrial DNA within these biopsies, and that's a kind of commonly used, albeit somewhat crude marker of the amount of mitochondrial DNA and is linked to mitochondrial biogenesis, which again I'll come back to. We, a few years ago, did a relatively small study using phosphorus MRS in skeletal muscle in HIV. And if you look at the right-hand panel here, first of all, this is the HIV-positive individuals, and you can see that there are a handful of them who had this slow recovery suggesting impaired mitochondria. What was more surprising to come out of this study, though, was that if you just look at their resting state in the scanner, something called their phosphorylation potential, which is essentially the amount of ADP required to make a given amount of ATP. So, I mean, it's basically a measure of the resting or the homeostatic health of the mitochondrion. This was actually impaired in the HIV positive group. So this can potentially be a useful tool. But clearly these MRS studies, they're pretty expensive to do. Muscle biopsy studies are invasive, so they're only ever going to be relatively small studies. They're not suitable for very long follow-up periods. So this is a study that tries to get at this from a different angle and address some of those. So this was a large study from Hopkins a few years back. And what they've done is they've simply measured mitochondrial DNA levels, which I've already mentioned, in peripheral blood. They've divided individuals into quintiles based on how much mitochondrial DNA they had, and then they've actually followed them up for mortality. So it's a very hard endpoint. And you can see in both white and black individuals that the lower your mitochondrial DNA copy number in blood, the worse your mortality. And they also replicated this in another cohort. Now, on the one hand, I think, yes, this is kind of an expected result, making a link between mitochondria, frailty, and aging. On the other hand, my research is predominantly kind of mechanistic, as Julian said, and it's not absolutely clear to me where these changes in blood fall on that kind of mechanistic pathway, and that's something we're actually trying to think about at the moment. So we've mentioned that getting at causality in any kind of human aging study is very difficult, so that is potentially where animal models come in. I don't know how old people would have to be to grow up on Mighty Mouse. Did anyone watch Mighty Mouse? Yeah, David's nodding, okay. So, quite a few years ago now, there was a seminal paper which described a new mitochondrial aging mouse model, and this really took the field forward a huge leap. So this mouse has a mitochondrial polymerase, so the enzyme that replicates mitochondrial DNA is defective, and as a consequence, this mouse gets lots and lots and lots of mutations in the mitochondrial DNA. And as you can see, it has a very, very striking kind of aging phenotype. It's kind of hunched and thin and thin hair and so on. It's kind of almost too good to be true, really, actually, as a phenotype in a mouse model. And it also has a significant reduced life expectancy, so they tend to die just before a year of age. So that seemed to very much kind of almost wrap up this question of were mitochondria causally related to aging. Of course, things are never quite that simple, and then there was a series of papers that came out in the following years which kind of nuanced that a bit. So if you look at, first of all, at the turquoise bar here, so this is the total burden of mutations in this mouse model, and as you can see, it's sky high. If you compare that to the burden of mutations in an aged human, this is colon, but it doesn't really matter. I've lost my pointer now. In the blue, you can see that's a lot lower. And then in contrast, if you look at the green, that's the heterozygous mouse, which has an intermediate amount of mutations, but appears to age normally. So how is it that a low level of mutations might be in some way related to aging in a human, and yet a higher level isn't in a mouse? And you can certainly see these blue cells, this is in colon here, are abnormal, so something has happened to the mitochondria. And the answer to this is, at least in part, probably this concept called clonal expansion that I mentioned at the beginning. And this is something that I kind of do a lot of work on trying to understand mechanisms of, along with a number of others in our centre. It may sound a bit dry, but bear with me. So this is studies sectioning through single muscle fibres from aged individuals. And the portions of fibre that are blue here are abnormal in terms of their mitochondrial function. And this is a map at the bottom of a single fibre, and you can see there's a segment that's abnormal in the middle. And then if you do single-cell studies to map the molecular damage in this fibre, you can see that the levels of mutated mitochondrial DNA are very high here in the abnormal section and otherwise very low. So somehow this mutation has gone from a very low level to a very high level here. And understanding why this has happened is one of the things we're working on. Just as kind of an aside to bring back in the HIV, so this is absolutely what we think may be happening in individuals who've been exposed to some of these older NRTIs that we associate with mitochondrial toxicity. So a few years ago we did a study where we demonstrated that yes, they have these blue cells. When you look inside them, they have high levels of mutations. This is just a cartoon mapping where we saw those mutations within the mitochondrial genome. And then we did a computer study, modeling study with David Samuels from Vanderbilt, some of you may know, and this suggested that a period of exposure to these drugs could cause an increase in this process of clonal expansion. So this is now bringing this question right up to date, and Amy Vincent is a postdoc in our centre, and this is some data she's just published. So these images are an immunofluorescence technique that we've now kind of moved over to for looking at muscle and other tissues. In this one, blue is normal and red is abnormal, just to confuse you. I want you to look at panel B here, and hopefully you can appreciate there's a small but very intense pink dot here. And what I believe she's identified here is the kind of nascent event in clonal expansion. So this is an area where a mutation has arisen in a cell and is starting to then expand throughout the cell. So what we're doing now is studies to look at the molecular pathways, the signaling pathways that might be driving this event, and we're comparing what you see in HIV, in aging, in mitochondrial disease to see whether these are conserved pathways. So the next question I'm going to pose is, when does mitochondrial aging start? And that may seem a slightly esoteric question, but if we come back to this mouse that we've already met, so he starts to look ropey from maybe 4 months of age, something like that, so well into mouse adulthood. However, if you look at some of the replicative tissues in the mouse, you can actually go right back into the embryonic period, and there is already a defect. So this little excerpt down the bottom here, these are cells taken from mouse liver, and the capacity of these cells to self-renew has been tested ex vivo. And as you can see in the mutant mouse, they basically can't self-renew. So they've already lost their stemness, their ability to function as stem cells. So we clearly believe that loss of stem cell function, loss of this regenerative capacity is very important in human aging. But where mitochondria come into that and how early that might come in, that has very much not been figured out so far. So I mentioned free radicals at the beginning, and I'm just going to spend a few slides on this because this is perhaps one of the areas where how we view things has changed most in the last 5 to 10 years. So the free radical theory of aging is actually more than 60 years old now, which is quite unbelievable, really. And as far as mitochondria goes, it basically goes like this. So mitochondria are, of course, the source, or the main source of reactive oxygen species within a cell. They may damage molecules in their immediate vicinity, which will include the mitochondrial DNA itself, and that may set up a vicious cycle of damage to mitochondrial DNA, free radicals, more damage, etc. So that's quite a persuasive theory. Now, if you think about it, anything where you have a kind of positive feedback loop like that should result in an exponential increase in that parameter, so in this case, aging. So of course you can test, do you see that? And The bottom line, of course, is actually no, you don't. So this is data from Laura Greaves, again from our centre in human colon. The different colours of bars are different ways of attempting to measure the burden of mitochondrial DNA mutations. That's a story for another day, but the bottom line message is that you really don't see a significant, or certainly not an exponential upward trajectory. It's either very, very slow or almost a flat line. But clearly, these reactive oxygen species are there. So what is it that they're doing? So this is a cartoon from Yael Passos from Newcastle, who works on the interrelationship between cellular senescence and mitochondrial dysfunction, and has shown that induction of a senescent phenotype in cells is very dependent on mitochondria, and he believes that mitochondria are essentially acting as signaling molecules, driving some of these cells towards a senescent phenotype. And if you were to kind of summarize a lot of the work that's been done on ROS in the last 5+ years, it's kind of moved away from the concept so much of ROS as damage, which is what's kind of easier to measure, but in terms of the functional effect, it's almost certainly its role as signaling molecules that is more important. Now, just to stress, those signaling roles are not necessarily always detrimental. So I think we kind of, someone touched earlier on the kind of acute inflammatory response you might get after a bout of exercise. Well, you also get a kind of surge in ROS after a bout of exercise, but that is almost certainly a beneficial signal that helps bring about the adaptive response to exercise. So I'm going to finish off by talking about some of the kind of interventions that may be applicable in the field of aging in mitochondria. So this diagram is showing the important pathways that mediate mitochondrial biogenesis. So this concept basically is very simple, really. It's that if you can increase the number of mitochondria within cells, then you should be able to increase the energy production and therefore the health of those cells, and hopefully therefore the health of the individual. You hopefully appreciate that most of these pathways converge on this molecule here, PGC1 alpha, in the center, and this has been termed the master regulator of mitochondrial biogenesis. I'm sure you're probably aware that, at least in terms of human studies, that exercise is probably the most promising intervention out there in terms of improving mitochondrial function. This has typically been endurance exercise for mitochondrial function, although there's now some more recent interest in high-intensity exercise. And you can see that, one of the main ways this signals is, I've lost my pointer, but this seems to signal through, I've lost everything, okay, good. Yeah, so this seems to signal through PGC1 alpha. I'm just going to show one slide, however, that's just advanced our understanding of this mitochondrial exercise response, and I think this was from a Canadian group published just in the last few weeks. So they took sedentary individuals, young and old, and they gave an exercise intervention. And yes, they both saw some improvement in mitochondrial function, as you would hope, so that's great. But what was slightly concerning was there seemed to be some degree of essentially metabolic block or signaling block within the older people within their mitochondria. And the kind of two molecular features of this block that they picked up, one was that there seemed to be a bottleneck of ADP rather. So their ability to take ADP and push it through the mitochondrial respiratory chain to make ATP of energy seemed to be impaired. And then in parallel with this, they got a hugely increased amount of ROS production for the same degree of exercise challenge. So, and this imbalance of ADP and ATP is potentially interesting because that comes back to, well, that's a parallel, an analog of what we were measuring on that MRS study I showed you several slides ago. So, of course, what most of us might want is an intervention that replicates exercise without the hard work. So, and you know, there are lots of different things that have been tried for that. But perhaps I think the pathway that's looking most promising, or indeed is most kind of sexy at the minute, is this pathway here, which starts with NAD. It goes through SIRT1, so you maybe have heard of sirtuins. These are these molecules that are thought to be important in mediating the effect of caloric restriction, for example, so in these longevity studies of small animals. And again, that converges on PGC1 alpha. And the approach here, or one of the approaches, is very simple. You just kind of flood the system with extra NAD plus at the top, and that basically cranks the hose right open and hopefully pushes you through to more mitochondrial biogenesis. So this was a really interesting mouse study from Johan Auverg's group in Switzerland a couple of years ago. So what they did is they took aged mice. They gave them a compound called nicotinamide riboside, which increases the levels of this metabolite, NAD+. And you can see that they have a modest but significant increase in lifespan, which is very encouraging. But what was also particularly interesting to me, and thinking back to what we were talking about a couple of slides ago, is a lot of this benefit seemed to be from rescuing mitochondrial function within stem cells. So this is muscle from these mice at the bottom here, which have been subjected to an injury in order to try and make the muscle fibers regenerate. And in the normal aged mouse, basically, there's next to no regeneration that happens. Whereas in the mouse that's had this supplementation to its mitochondria, all these green fibers are ones that are actively regenerating. And in order for muscle fibers to be able to regenerate, there must be an intact stem cell population there. It will not surprise you to know that you can buy this stuff online. So if you would want to live a long and healthy life, you can get free shipping in the US only. So there you go. Okay, so I'm gonna just kind of wrap this up by just reiterating a few of the points I've made. So given a huge topic, I've just chosen to focus on about three things that I think are kind of quite topical, where our view has changed, as I said. So first of all, thinking about mutations in mitochondrial DNA. So we've kind of moved from this view that they just ramp up and up and up over life to this view that actually perhaps the total burden doesn't change so much, but it's this kind of stochastic effect on individual cells that gradually ramps up over time. ROS, so probably we should now be thinking in terms of much more about the signaling capacity of ROS, and what's really interesting is how this then interplays in cellular senescence. We always used to start mitochondrial talks by saying how mitochondria are really important in cells like neurons and muscle, which need lots of energy, they're post-mitotic, blah, blah. So that's kind of all true, but it actually turns out that maybe we've been wrong to disregard post-mitotic tissues, because actually it seems like mitochondria are going to turn out to be pretty important in the maintenance of stem cell function in those tissues. And then finally, bringing this back to HIV right at the end, so trying to think, are there specific aspects of HIV or antiretroviral therapy that might then have an additional effect on some of these parameters? So I think the effect that some of these older NRTIs might have on mitochondrial DNA mutations is reasonably well established by our group and others. I think the effect that more modern antiretrovirals, or indeed residual virus itself, might have is still an open question. One of my students, Matt's going to tell you a little bit about that in half an hour or so. We do know that HIV induces some changes in ROS, as may some antiretrovirals, but really, to my knowledge, very little has been done on what then the downstream signaling effects of that are. And in terms of stem cells, so there's been a little bit of work, as you can imagine, on immune senescence and hematological stem cell populations, but very little beyond that in HIV, and certainly very little that tries to look at what the role of mitochondria is in that. So there's plenty to write grants on if people are so inclined. So I'm going to wrap it up there. I promised Julian I'd finish within time, which I think I have. So just to thank and acknowledge our funders and various collaborators. I'm very happy to take questions. Thank you.